This is part 2 of the genomics series. This part's on RNA viruses, and you can find a lot of the material in chapter 27 of Integrated Molecular Evolution, the second edition. The RNA viruses are way down here on this chart. They have among the smallest genomes, and of course they're obligate parasites. So I'll start by talking about HIV, and then go on to influenza A, and then Ebola, and finally coronaviruses. These are scanning electron micrographs and transmission electron micrographs of HIV. Here they are coding a T cell on the top over here. You can also see the tiny green particles. Those are the HIV particles on the cells. These are false colors, so they're not colored green. And then the transmission electron micrographs show the interior of those viruses. So you can see roughly a spherical particle, and then inside is the capsid of the virus itself, where the RNA and a couple of protein molecules are housed. So here's a diagram of the human immunodeficiency virus, HIV, which is an RNA virus of Baltimore class 6, so it has a reverse transcriptase that it carries along with it. Causative agent of AIDS, acquired immunodeficiency syndrome. The virus on the outside has glycoprotein 120 that attaches, is recognized by the CD4 host cell receptors, which is where it connects with the T cells. Then there's GP41, which is a transmembrane protein. Those two are actually made as a polyprotein and then broken up and reassembled. Then there's the host membrane in red, so that's from the previous host cell that was infected. The GAG gene produces the matrix protein, which fills up the interior portion of the virion. It also encodes for the capsid protein, which surrounds the chromosomes of the virus, as well as a polyprotein. So there are the two RNA molecules, they're identical copies. And then the pol is carried along with that. That's another polyprotein, which contains the protease, the reverse transcriptase, RNase H, and an integrase, which aids in integrating the DNA copy of the virus into the host chromosome. Here is the sequence of HIV. The complete sequence is 9,181 nucleotides of RNA, although there are variations on this total length for different strains of HIV. And it's always surprising to me that you have a virus that's with a very small genome, only 9,100 and something nucleotides, but it can take down and kill a human that has a genome size of 3.2 billion nucleotides. Here's the life cycle of HIV. Of course, it can be transmitted through bodily fluids, either by sexual means or through blood transfusions and secretions. So the first thing it does is it attaches to the CD4 receptors on T lymphocytes. That's the GP120 protein that attaches. And it fuses with the membrane and then starts replication, production of new virus particles from there. So it can go through repeated cycles, infecting more and more cells. Many times people infected with HIV will experience flu-like symptoms, although some people are symptomless and are surprised when they find out they come out HIV positive. At some point in time, in some cells, you'll get integration of the DNA copy of the RNA, the viral RNA, into the lymphocyte chromosome. It can be triggered to go from this lysogenic stage into the lytic stage and also produce more viruses, and this can happen continuously. Later on, the virus will start changing the genome of and control of the genome of the lymphocyte and make it into a multinucleate syncytium. So it will have absolutely no immune response, which is usually what eventually causes the death of the patient, not the HIV itself, but the lack of an immune system. So you can get also from the syncytium, it becomes more or less a virus factory, producing more and more virus particles, and those go on to infect more and more T cells, which are a central part of the acquired immune response. This is a graph of disease progression. You can see at time 0, you get a primary infection, and there's quite a high CD4 T lymphocyte count. So it's reacting against the virus, and then you have an increase in the HIV viruses, and so you'll start having, after several weeks, a decline in the CD4 lymphocytes, which are becoming infected and being taken over by the HIV control mechanisms now, producing more virus particles. Then there's a latent period where there's a decrease in the amount of virus, an increase in the number of CD4 cells, so it looks like there's an immune response to knock out the virus. But then slowly, over a period of years, the CD4 cells continue to decline as they're taken over by the viruses, and you have an increase in the virus particles. Eventually, opportunistic infections take over as there's more and more failure of the immune system, and eventually death. Although there are quite a few drugs now and drug cocktails, that is 2 and 3 drugs at a time are taken, and so people are surviving more long-term now than they did in the past. Here's a general picture of an HIV virion. The GP120 is the surface protein that interacts with the CD4 receptors. The GP41 is the transmembrane portion, and you can see there are trimers of each of those proteins going through the membrane, which is part of the host membrane that they picked up on their way out of the cell. And then inside of that is the matrix protein, fills up the internal portions of the virion. And then inside of that is the capsid protein that surrounds the 2 copies of the virus genome. And coding those is another protein, nucleocapsid protein. And also a reverse transcriptase, and it's actually a polyprotein, as I mentioned before, containing the reverse transcriptase, a protease, an integrase, and RNase H. Here are the genomes of both HIV-1 and HIV-2. They're just slightly different versions of one another. The first thing to notice is they have 5' and 3' long terminal repeats, LTRs, and so this is like LTR retrotransposons. The GAG gene encodes for the matrix protein, the capsid protein, the nucleocapsid protein, and one other protein, P6. POL encodes for the protease, reverse transcriptase, and integrase. The ENV envelope gene encodes for the surface protein and the transmembrane protein on the outside, and then the others are to control replication and construction of the virion itself. As I indicated before, RNA viruses mutate at much higher rates than do DNA viruses. So this is actually a phylogram of HIV genomes throughout the world, and you can see that there's a great deal of variation in those different strains, and you can already see that from the fact that there's an HIV-1 and an HIV-2, but even each of those is quite variable as well. So although there's a great deal of variation throughout the world, you might be surprised by this one, because these are different sequences that were obtained from a single individual human who had HIV, and these samples were taken at different times, varying over several years. So you can see even within a certain individual, you are getting mutation and evolution of this virus, and that's why sometimes people will take drugs and the drugs will work for a while, and then they'll start failing, and the virus starts to replicate again and become a problem because it's evolving resistance to those particular drugs. So this is a diagram of the infection life cycle for this virus. So the GP120, the glycoprotein 120, attaches to the CD4 receptors on the cell, which are in the T lymphocytes. And then the RNAs are, plus the polyprotein is released into the cell. The reverse transcriptase starts copying a DNA copy of it. Those are then mobilized into the nucleus. You get expression of the genes, and then those mRNAs wind up out in the cytosol again, and you get ribosomal translation followed by encapsulation of the new RNAs and the polyprotein, which contains a reverse transcriptase. So integration occurs also back in the nucleus, and then that cell becomes just a factory again for producing new HIV virions. This is a slightly different version of the same thing, just showing a few of the additional details. So the first thing you get is attachment to the CD4 receptors on the T lymphocyte, release of the capsid, and then release of the RNA and the integrase that proceeds to the nucleus, and you're getting reverse transcription at the same time. You get integration of the chromosome as well as transcription from the viral DNA copy. And then those copies go out into the cytosol. They're translated on cytosolic ribosomes, and you're also getting copies of the virion chromosome being produced. That's all winds up going out to the membrane, and then you have new virions encapsulating the capsid with the RNA and the reverse transcriptase, etc. Finally, that a complete virus particle is then released from that cell, carrying with it a piece of the host membrane as well. So the next RNA virus I want to discuss is influenza A. It's in Baltimore class number 5, and you can find it in many shorebirds and waterfowl, and so those are natural reservoirs for influenza. And we already know that there are 18 what are called H types, hemagglutinin, for the hemagglutinin protein, and at least 11 neuraminidase types. So you put those together, and there are many, many different combinations of those. They then enter domestic birds from the wild avian hosts. Those can be transmitted to pigs and then eventually to humans. So the host range is quite broad. So the native hosts can carry from H1 to H19 and N1 to N11. So a pretty varied set of strains of influenza. They can be transmitted, some of them can be transmitted to humans, including H1, H2, and H3, and occasionally H5, H9, and H7. But many of them can infect domestic animals, including chickens and cats, horses, and pigs, and cattle, but they can also infect seals, whales, and other mammals and birds. This is a phylogram of the influenza strains based on their hemagglutinin subtypes. So you can see H1 through H18 here. The ones that have the circles around them, H1, H2, and H3, those are the primary strains that can infect humans and be transmitted from human to human. The other ones in triangles, H5, H7, and H9, are mainly avian types, but they can occasionally be transferred from the bird to the humans, and in that case, they're fatal quite a bit of the time. This is the typical ones that you'd call bird flu, and it's H5 was the one that was such a worry, and it's still circulating today, but it was such a worry starting in the mid 2000s. The people that most often contract H5, H7, and H9 are people who own their own birds, so that they're butchering birds and getting some of the blood on them and transferring it to themselves. Another population of people that often contract the bird flu are veterinarians that deal with injured and sick birds. So regarding the virulent bird flu, H5N1, it actually is highly virulent in birds, humans, and swine, so it's a pretty deadly one. Most influenza A strains are primarily mild intestinal viruses in birds, so they cause minimal disease in the birds. It's only in some cases where they attack the lungs and liver and other tissues that they become quite deadly. So the ones that attack the epithelia of our lungs really cause the most damage, and people end up suffocating in their own fluids, actually. Birds, humans, and other animals can die then from asphyxiation. It can become systemic too, so you can get brain damage and liver failure as well. This is a phylogram of H1 subtypes, and you can see there's quite a bit of variation even among H1 subtypes. It's been rooted to H6, which is the next group out from this one. That clade at the top contains the H1N1 strain that was the one that caused the 1918 pandemic. And the other groups down here are ones that cause more mild disease, not pandemic flus. And the ones here that says lake ice and lake water, those are ones that we studied, and I'll talk a little bit more about those. But first, a little more about the virus. It actually has 8 chromosomes, so it carries 8 pieces of RNA coated with nucleoprotein. It also carries along with it an RNA-dependent RNA polymerase. Then there's the matrix protein. The phospholipid bilayer comes from the host membrane. And then there's the hemagglutinin, which is a trimer, and the neuraminidase, which is a tetramer. An M2 ion, which is an ion channel, which is necessary for release of the virus inside of the cell. And here are some pictures of influenza A taken with a transmission electron microscope. And you can see that they take on a variety of shapes. Here are the genes on each of the 8 chromosomes. So PB2 encodes for the transcriptase and cap binding of the mRNA. PB1 encodes for transcriptase, as well as the PB1F2 for cell death, and it inhibits mitochondria. The PA is also for the transcriptase and the protease. HA, that's the hemagglutinin surface protein. NP is the nuclear protein, RNA binding and RNA transport. NA is the gene for the neuraminidase protein. And then the M1, M2 are those matrix proteins. And NS1 and NS2 are for non-structural proteins that have functions such as RNA transport, splicing, translation, and export of the viral RNA from the nucleus. If you total up the lengths of all these, they add up to 15,000 nucleotides. So as you can see, it has 8 different sections or 8 chromosomes that encode 8 sets of different proteins. And these can, if you get mixing of 2 or more of these strains into a single animal, such as a swine, that's usually what's called the mixing bowl, you can get what are called reassortant influenza strains. So in some cases, they can carry pieces that have a swine origin or a human origin or an avian origin altogether. And this is what the 2009 H1N1 was. So somewhere back in around 2000, there were some reassortants that had H3N2 and H1N2, and the origins of the fragments were from swine, human, and avian. And then in 2009, these mixed with a swine, a Eurasian swine strain, and became the 2009 H1N1. This one caused a lot of morbidity and mortality. Actually, I got this one, and it was a rather nasty one. So I had a temperature of 104 for 2 days, and then it went down to 102 for another 2 days, and then ended with a cough that lasted quite a while. But you can see that it contains some purple or some fragments from avian origin, some from swine origin, and some from human origin. And in some cases, when you get these reassortants, then they can be quite deadly because they can attack a population that is not resistant to them. You also can get recombination occurring for each one of those chromosomes as well. So there's quite a bit of variability in influenza, and this is why every year it's difficult to tell exactly what strain, what combination of genes, reassortants, recombination events are going to appear that year. Here is the infection cycle for influenza. The hemagglutinin protein on the outside of the virion interacts with sialic acids, which are sugar groups on the ends of proteins on this cell membrane of the host cell. The virus is then enveloped into an endosome, and then the M2 ion channel acidifies the endosome, and that causes release of the virions as well as the proteins, the RDRP, into the nucleus. And then you get the RDRP making mRNA strands and new viral chromosomes. Those then move back into the cytosol. Those are enveloped into new budding virions and eventually released from the cell. Now you'll note on the right-hand side that the proteins no longer have the sialic acid because the virus actually clips those off so that the newly released particles don't stick to the same cell but are released and then can stick to other cells with intact sialic acid residues. In the past, it had been noted that strains of influenza tend to recur over a certain number of years, usually corresponding to a time when there's no longer herd immunity in the human population. And so in 1977, there was a Russian influenza that looked very similar to the 1918 influenza. And then in 1968, there was a Hong Kong flu that looked similar to a strain that was present in the 1900 epidemic, and then an Asiatic 1957 Asiatic flu that looked similar, at least in its infection and death rate, to an 1889 influenza. So we and others think that there might be these strains recirculating over time. So I can explain this a little bit. We came up with an idea called genome or organism recycling, wherein organisms are entrapped in glaciers or environmental ice or lake ice. And then at a later time, they melt out. They then infect shorebirds and waterfowl. And those are then carried to the population. And some of those become deposited again during the winter or on top of glaciers to be frozen again and then come out at a later time. So in this case, there's a biotic cycle that's in migratory birds, especially shorebirds and aquatic waterfowl. And so they pick up the virus in the summer, in the spring and summer, up in their grounds where they reproduce. And then in the fall and the winter, they fly south, taking those viruses with them, and then return again the next summer and drop those off again. But we and others have shown that there's also an abiotic cycle in that in the summer, you have the birds essentially depositing these viruses in the water of the lakes and streams where they're reproducing. And this water is frozen during the winter, and it actually preserves these viruses quite well in some cases, including influenza. And then in the spring, when they start to melt, they release the viruses again. The birds are coming back, so then they're infected with usually a different virus than what they picked up the year before, or at least a different strain. We were lucky enough to get some samples from Siberia, from 3 lakes that freeze in the winter. Actually, they're frozen for about 6 months of the year, and then thaw out, and they're areas where birds come back every year. So there's Lake Adoma, Lake Shichichi, and Lake Park. Both Lake Adoma and Lake Shichichi are close to human habitation, and so the birds usually don't stay there very long, and they don't reproduce as much, whereas Lake Park is very remote, and there's a lot of young chicks around there, so they reproduce a lot to a much greater extent around Lake Park. This location is interesting because it coincides with several bird migratory routes, the East Asia as well as the Pacific routes. Quite a few birds use this, and there's a lot of mixing during the year between different birds arriving and different birds leaving at different times and depositing things in the lake. So we were interested to find out the sequences that we could find up in those lakes and whether those had any relation to any strains that had appeared in the human population over the years. So this is the sequences we got were here in both lake ice and lake water, primarily from Lake Park. I think we only got one sequence from the other 2 lakes, but we got several sequences from Lake Park. And interestingly enough, in the cladogram or the phylogram, they came out very close to a couple of strains, one from Tokyo that was from 1933 and 1967, another group that were found in 1939, and then some others that were around in the human population in 1938. So we think this is evidence for this genome recycling over a period of many decades. So I've talked about two RNA viruses so far, HIV that has about a 9,000 nucleotide genome, and influenza A with about a 15,000 nucleotide genome. So next I want to talk about Ebola and then coronavirus. Ebola is a negative sense RNA virus, first described from the Ebola River in Africa. But it attacks cells in such a way that the cells kind of break open, release all of their internal parts, and pretty much just liquefy tissues, including liver and things like that. So people will bleed out of their eyes and ears and different places on their body as their tissues liquefy. It has a fairly simple form and a fairly simple life cycle in that it makes new copies of itself, forming mRNAs as well as a plus strand of the RNA. Minus strands are made of that, and the proteins are put together into new virions. And it never enters the nucleus, so this is all cytosolic. So Ebola and its relative, Marburg virus, very similar. So these diseases have cropped up in Africa as well as Europe. In fact, at least one of the things that was called a plague in Italy is thought now to possibly have been Ebola brought back from Africa from sailors on a ship that had just gone to Africa to pick up some goods. So you can see there's a nucleoprotein and several other viral proteins, VP35, VP40. The GP is a glycoprotein, that's the peplomers on the outside, and then the L there is the RNA-dependent RNA polymerase, the RDRP, that's used by the virus to make new copies of itself. There's the form of the virus, the peplomers on the outside that interact with the cell surface proteins, the envelope proteins, matrix protein, nucleocapsid, and RNA. So fairly simple viruses. Ebola and Marburg have slightly different forms, but very similar genes. In total, the genome size is about 19,000 nucleotides. So again, fairly small genomes that can bring down a human. The last group of viruses I want to talk about are the coronaviruses. Here's one of the virions split open here. There's the spike glycoprotein, which attaches to cell surface proteins on the host cell. And then the M protein, the envelope, which is a membrane that is not from the cell surface, but from the endoplasmic reticulum, the membrane of the endoplasmic reticulum. There's the E protein, which I'll talk about a little bit, and then the RNA inside. These have large genomes for RNA viruses, about 30,000 nucleotides or so. This is a transmission electron micrograph of some of the virions, where you can clearly see some of the spike proteins sticking out of the virion. Here's a recent table for this coronavirus, which is the SARS-CoV-2 virus that causes the COVID-19 disease. And you can see that as people are older and older get infected, the fatality rate goes up by quite a bit. The overall rate is 1.8 to 3.4, and this is within the range of the 1918 influenza death rate. Part of this is because they cause very similar reactions in the body. So it's not the virus that's actually killing the people. The viruses are causing a heightened immune response, especially part of the innate system, which causes inflammation and fever and local cell death. So what's actually happening is you're getting inflammation, damage of the lung tissue, release of fluids, and people have trouble breathing because they're essentially drowning in their own fluids. Again, as with all RNA viruses, they mutate rapidly. So in 2020, during the pandemic, scientists have been tracking 8 strains of the SARS-CoV-2 virus, which causes COVID-19. 19 because it's 2019 was when it was discovered. And so, although it may have started in China, it mutated. You can have a whole bunch of a population of people with the virus. Some of those mutate, they move, and so they're taking those mutants with them. That then gets established in a new area. So you can see some moving from China into Europe, moving from China into the U.S., moving from Europe into the U.S., and then into South America and other locales around the world. Here are the infection cycles for both SARS and MERS, SARS being the severe acute respiratory syndrome, and MERS, the Middle East respiratory syndrome virus. They both become enclosed in a membrane structure, and then they start translation on cytoplasmic ribosomes. They create many, many different RNAs, which I'll show you in a few minutes. Some parts emerge from the endoplasmic reticulum, including the surface proteins, and those are then modified in the Golgi, and then they wind up being part of the membrane of the Golgi. The genomic RNA is then put into the inside of those vesicles, and the vesicles are then released from the host cells. So the membrane that surrounds these virions starts as a membrane from the ER, and then becomes part of the Golgi also, and so that becomes the membrane that surrounds these virions. The surface proteins, specifically the spike glycoprotein, sticking out of the virion, are the things that interact with the host cell specifically, so that they become attached to those host cells and then infect them. So here are some of the genes. These are the genomes of SARS-CoV and MERS-CoV, and you can see that they're very similar, although they have a few differences in their structure. So this first gene encodes a protein that is responsible for proteolysis. It's also an interferon antagonist, so it interferes with some of the immune responses, and it also is involved in other functions in the cell to identify and destroy proteins. And then there's this middle gene, ORF1b, which encodes the viral replication and transcription proteins. And then the S gene there, that's for the spike glycoprotein. And then some of the other genes at the end, EMN, and some of the other pieces there are also surface proteins to maintain integrity of the virion, as well as interact with the immune system to disable it, essentially. So some of the functions of this virion, the coronaviruses, they stimulate some interactions of the immune system while they inhibit other functions of the immune system. So this diagram shows a bit more detail of ORF1A and ORF1B, as well as some of the other genes and proteins. So the single protein that's made is a polyprotein that includes the first about 60% of the genome, and it makes different proteins, including a proteinase and proteins for transcription and replication, and the RDRP, that is the RNA-dependent RNA polymerase. Polyproteins are common in many of the RNA viruses. And so a polyprotein is one long protein. Often it includes a proteinase, and the proteinase acts to clip up the protein into its various forms. So part of this protein makes the RDRP, the proteinase, a membrane-bound protein. Next down the line is the S gene, which encodes the spike glycoprotein. That's clear. That's what interacts with the host cell membranes. The other ones down below, the RNA 3, 4, 5, encode some membrane-bound proteins as well as some other proteins, and some act as just RNAs. Those affect virion infection, interfering with parts of the immune system, as well as part of the assembly of the virion as well. Here's a different view of the genome of SARS-CoV and MERS-CoV. Similar to each other, but some differences, and there's a special emphasis on the spike glycoprotein here. So there's two subunits, S1 and S2 in each case, and on the amino end there's a signal peptide, which then makes that associate with the endoplasmic reticulum and Golgi. And then on the other end, you see the TM region, and that's the transmembrane portion that fits into the membrane. And then all of the other parts above that are sticking out into the outside portions of the virion, and specifically the RBD, RBM regions. Those are the portions that interact with the surface receptors on the host cells. Okay, that's all I have to say about RNA viruses. I will talk next time in the next presentation about DNA viruses and some details about expression of their genes. Thank you very much.